Okay, so welcome to the final video for Module 10, where we're focusing on DNA, RNA, transcription, translation, and processes that can modify and change the information in DNA. And that's really what I want to focus on in this final video, so over the next few minutes. So basically, looking at how DNA's nucleotide sequences can be changed. And any change that you get in those specific nucleotide sequences is referred to as a mutation. Now, mutations can be very simple, where they just are changing a single nucleotide, or it can involve very large regions of a chromosome where you have large regions being changed and modified. Mutations within a gene can basically be divided into two general categories. You can have nucleotide substitutions, where you're basically switching specific nucleotides, or you can insert a new nucleotide, or you can delete a previous nucleotide. And each of those individual categories or individual processes have very different effects, or could have very different effects on the resulting genetic sequence and the final protein amino acid sequence that results from that initial genetic sequence. So here we have a normal hemoglobin DNA sequence, the associated mRNA sequence, and the associated polypeptide. This particular codon, CTT in the DNA, or GAA in the mRNA, codes for a specific amino acid. Now, if this T is substituted for an A, you get a corresponding substitution for a U in the mRNA. So now your codon is not GAA, instead it's GUA. And remember, we said that if you get changes at the first or second codon, that usually ends up with a change in the amino acid, whereas sometimes if you end up with a change at the third codon, you could still get the same amino acid. In this situation, we're changing that second codon, so we are getting a change in the amino acid. So we're going from glutamine to valine. And so you're getting a specific change in this protein. So this one change completely changes the function of the hemoglobin protein. So you go from a normal hemoglobin to sickle cell hemoglobin. So sickle cell disease appears just from this one mutation, which is pretty crazy, that just one simple change can lead to a disease that can really affect the human body. Now, there are a couple of different types of mutations. So you can have what's called sense mutations that involve a single nucleotide and change the amino acid coding. Okay? So for example, with the sickle cell example we looked at in the previous slide. Nonsense mutations change an amino acid codon into a stop codon. So basically with nonsense, you get, say, a working sequence of codons, but all of a sudden you have a mutation that inserts a stop codon, say, right in the middle of that amino acid sequence. So now all of a sudden your protein sequence, your amino acid sequence is a lot shorter and stops a lot sooner than what it initially should have. Other types of mutations, such as deletions and insertions, will lead to one or more nucleotides in a gene, and that can either shorten the sequence or lengthen the sequence, and it leads to what we call frame shift mutations, and are often very disastrous, and they often are very deleterious, which means they have a big negative effect. If you add or subtract nucleotides, you're altering the codon pairing. So basically you're altering the triplet grouping of your genetic material. And so a frame shift mutation often produces a protein that doesn't function anymore. So here we have these three types of mutations being shown in this figure. So here you have a base substitution where you are changing the amino acid sequence, right? So you're ending up with what was initially a glycine to a serine, and so that's a missense mutation. This deletion of the U nucleotide here, so that triple U nucleotide gets deleted. So that third U gets deleted. Now instead of having triple U, your codon is UUG. UUG codes for a completely different amino acid, which is leucine. But you're also shifting all of the downstream codons. So now instead of UUU, AGC, GCA, you now get, or I'm sorry, UUU, GGC, GCA, which is the appropriate RNA nucleotide codons. You now get UUG, GCG, and CA. So the last two nucleotides don't even code for anything anymore. So not only are you shifting, you're also cutting off. You're one amino acid short. If you insert, you can shift everything downstream as well. So now you UUG because of the insertion, and then you end up with UGG, CGC, so completely different amino acids, and all of that's shifted and affected downstream. So you're not just changing one amino acid that may not make a big difference. You're completely changing everything downstream of that deletion or insertion. And that's why they're so detrimental, because you're completely changing the back portion of that polypeptide sequence. And now just for a final information, so mutagens, which can occur in a number of ways, are basically just these spontaneous mutations that come from random errors in DNA replication and recombination. You can also have mutagens that come from substances such as radiation, which come from X-rays or ultraviolet light, and other mutagens can also act as carcinogens. So some of these mutagens may not be harmful, but other mutagens could cause specific types of cancer. And so that's where you see warnings. So this could potentially cause carcinogens, etc. And so that's what that exactly means, is that it leads to a mutagen that could potentially cause cancer. And so although mutagens are most often harmful, sometimes they can be beneficial. I'm sorry, not mutagens, mutations. So although mutations can be harmful and sometimes are, most of the time they're neutral or could even be beneficial. Neutral meaning they don't have a large effect. But whenever they're beneficial, they could lead to increases in diversity. They could lead to increases in variation of genes, to speciation events. Once you get species becoming different enough because of an increased accumulation of mutations that leads one population to be different from another. So mutations are not always harmful. Sometimes they don't have any effect. Sometimes they have a beneficial effect. And so it's important to understand how these mutations can affect the associated proteins, and then we can look at those associated proteins to determine whether or not the after effect is deleterious or beneficial. And so we're just going to end with this. So here's two questions to think about. So one, what would happen if a mutation changed a start codon to some other codon? And two, what happens when one nucleotide is lost from the middle of a gene? Okay? So take a minute to answer these. Okay, so for the first one, if you have a mutation to change from a start codon to another codon, you basically end up with a non-functional gene, because the polymerase doesn't, like the tRNA doesn't know where to start, doesn't know where the methionine should be placed, so you won't get a polypeptide being made. You have a non-functional gene. You won't have any translation being initiated. In this second case, you end up with a frame shift. So you end up with that episode of everything downstream of that nucleotide being lost, basically shifting, and you're going to have a long string of incorrect amino acids, most likely leading to a non-functional polypeptide or protein. Okay? And so that's it for Module 10, where we looked at DNA, RNA, the structure of both nucleotides, how you carry information from DNA to RNA to proteins, and then also how DNA is replicated so that that information can be transferred from parent to offspring. And so thank you for sticking around and watching the videos for Module 10. We will see you next week whenever we start Module 11. So have a good rest of your week or weekend.